right, well, I'd like to thank everybody for coming tonight, and I'd like to thank you for the invitation to speak to you tonight about the research I will be doing that was graciously funded by a grant from the Scottish Rite Charitable Foundation of Canada. The title of the grant that I have received is I'll explain this, it's called The Role of the Cellular Prion Protein in Alpha-Synuclein Propagation. That's a lot of big words, but basically it's about understanding how we can treat Parkinson's disease. So as mentioned in the introduction, I'm part of the Tanz Center for Research in Neurodegenerative Diseases, and as an institute, we have a collection of 10 individuals who are devoting their lives to studying and trying to find cures for these diseases of the brain, which include things like Alzheimer's disease, Parkinson's disease, Huntington's disease, ALS, which everybody knows of now because of the Ice Bucket Challenge, as well as diseases like frontotemporal dementia, which are becoming more and more apparent in athletes who have experienced lots of concussions, also called chronic traumatic encephalopathy. Now, these are diseases of the brain, and they're called neurodegenerative diseases because the common feature is that you're losing brain cells or neurons in the brain. And depending on where you're losing these and what parts of the brain, that's what causes the different types of disease. The other feature is that they all involve what's called protein misfolding. This is where a protein in the brain kind of clumps together with itself to form these large, big masses inside the cell that disrupt the function of a cell. Now, unfortunately, all of these diseases are progressive diseases, which means that they get worse over time. So unlike a disease like a stroke, where you might be expected to regain some function after a single adverse event, diseases like Alzheimer's disease and Parkinson's disease gradually get worse over time. And as of now, we have no cures for these diseases that address the actual causes of the disorders. So I'm going to be talking to you about Parkinson's disease specifically. So Parkinson's disease is the second most common of the neurodegenerative diseases of humans, next to Alzheimer's disease, of course. The incidence is about one new case per 5,000 people in a year. But if you look at the prevalence of the disease, which is how many people have it in the population at any given time, that if you are over the age of 70, basically about 1 in 100 individuals will have Parkinson's disease. So it's a very common disorder, especially as one gets older. Now, Parkinson's disease clinically is fairly easily recognized. It can be caused, these are the telltale symptoms. So a tremor in your sort of hand is often the most obvious sign, but also a general stiffness and slowness of movement, as well as instability in your posture. So if you look at the brain of a person who has Parkinson's disease compared to a normal brain, and hopefully most of you can see this. So on the left here is what a normal brain would look like, and this is a part of the brain called the basal ganglia. So what goes wrong in Parkinson's disease is here in this darker area called the substantia nigra, is you lose the cells in this region. So here is a normal brain. You have lots of the cells that produce a black pigment, which is why it's called the substantia nigra. And over here is a Parkinson's disease brain where you can see you've lost a lot of this black cells. And what this does is that these cells produce a chemical called dopamine. And dopamine is important for cells communicating with each other in the brain, which is important for how we move. And so when you lose these cells, you don't produce enough dopamine, and therefore the communication goes haywire, and you end up having a movement disorder. So Parkinson's disease is, in some ways, sort of the best neurodegenerative disease to have, if you have to have one, because you can manage the symptoms. So you can replace this loss of dopamine by giving a synthetic version called levodopa or L-dopa. I don't know if any of you here have seen the movie Awakenings with Robert De Niro. That's what that movie is about, how Parkinson's patients kind of become alive after receiving this. And so this treats the symptoms, but it doesn't actually address the root cause of the disease. And so the disease will continue to progress. But if you have Parkinson's disease, you can live 20 or more years, for example, somebody like Michael J. Fox. And if you ever see Michael J. Fox, and you'll notice that he has sort of uncontrolled movements, that is actually a cause of the levodopa treatment, not Parkinson's disease itself. So if you zoom in even farther and you look at the cells in the brain in a patient who has Parkinson's disease, you'll see what these big masses of protein clumps or protein aggregates called Lewy bodies. So these are basically things that take up space in the cell and disrupt the normal ability of a cell to do its job. And these Lewy bodies are made up of a protein called alpha-synuclein. This is what alpha-synuclein looks like. So one of these Lewy bodies would contain hundreds of thousands of copies of that protein all clumped together in a big mass. And these are thought to be what eventually kills the cells in Parkinson's disease. So alpha-synuclein is a very important molecule for understanding this disease. So now these Lewy bodies, these insoluble masses of protein in cells, don't occur at random places in the brain. If you look at very earlier mild cases of Parkinson's disease, you'll only find the Lewy bodies in what's called the brainstem region. If you look at cases that are a bit more severe, more advanced, then you'll see that there are also aggregates in the substantia nigra. And if you look at patients who have very, very advanced Parkinson's disease, you'll also find them in the outer regions of the brain. And so it's a very sort of predictable pattern, suggesting that maybe these aggregates, these Lewy bodies, can actually spread from cell to cell in the brain. And we think that this is why the disease eventually gets worse. As it spreads to more and more regions of the brain, you're infecting more and more functions of the brain, which turns into a worsening of your symptoms. So understanding why alpha-synuclein in these Lewy bodies can go from cell to cell is an important question, we think, for understanding the disease. So now alpha-synuclein as a protein is produced inside a cell, and these Lewy bodies are also found inside cells. So cells are kind of like your house. You want to keep the outside of your house outside so you have a nice, comfortable environment to live in. Same thing with cells. You can't just non-discriminately leave the cell or come into the cell without having a special way of doing it. So if alpha-synuclein is able to spread from cell to cell, there has to be some sort of mechanism that allows it to do that. So here you have alpha-synuclein, and it clumps together to form these Lewy body aggregates. And then what we think might be happening, and there is some evidence for this, is that there might be another protein that sits on the outside of a cell, so that once these aggregates get outside of a cell, there's something that sort of grabs onto them and takes them into the neighboring cell. And that can start off the disease process in that cell, and that it can go on and between neighboring cells in the brain. And so knowing what these sort of molecules are that can grab hold of these aggregates and allow them to spread, we think is really important for understanding the disease. And the molecule that I'm focusing on in my research is a molecule called PRPC. And so we think that PRPC might be sort of like the key that allows you to open the door inside the cell for alpha-synuclein aggregates in Parkinson's disease. So PRPC is actually a molecule that's involved in a completely different brain disease, a set of diseases actually, called the prion diseases. So I think most people probably have heard of mad cow disease, which is the most famous prion disease. So prion diseases affect both animals and humans. In humans, the most common disease is called Creutzfeldt-Jakob disease, or CJD. It's very rare. About 1 in a million people develop this disease. It's actually why I got into research, because my father was one of those unlucky 1 in a million people, and he died of Creutzfeldt-Jakob disease. So that's how I started doing research in the field of neurodegenerative illnesses. And so the other part of my time I spend studying this type of disease. So this diseases include, and so the interesting thing about prion diseases is that they have the potential, unlike Alzheimer's disease or Parkinson's disease, to be infectious or transmissible from person to person, or from, in the case of prion diseases and mad cow disease, from animal to human. So what exactly are prion diseases? So in prion disease, you have this protein, which I just mentioned, called PRPC. PRP stands for prion protein. C stands for cellular. So if you think, this is kind of an abstract version of the disease. So if you have this molecule called PRPC, it's a protein. So proteins normally have a shape, and that shape is important for what they do in the brain. So their shape sort of allows them to do whatever their specialized function is. And so proteins are dynamic entities. So if you think of PRPC as a Chevrolet Corvette, proteins, they can do slight movements. Their shape can change slightly. So if you think of opening the hood of a car, or opening the door, or putting the roof down. These are sort of relatively small changes that don't really change the overall shape of the molecule. However, the neat thing about prions is that prions can exist in two different shapes. And the other shape, which is called PRP scrapie, is very bad and dangerous. And this is the molecule that is actually causes these diseases to be infectious. So you can think of it in the analogy of a transformer. Now, the research I'm doing is on the normal version of the protein, not this guy. And so this protein, we all have it in our brains at very high levels, but there's a certain conundrum in that we don't really know why we have it. And it's quite high levels in the brain. A lot of different organisms have it. And it seems very unlikely that the only reason we have it is so that 1 in a million people can get prion disease. So it must be doing something useful in the brain. We're just not quite sure what it is yet. And so this is what this molecule looks like. And we think that maybe what it's doing is that it's important for cells talking to each other or bringing things in between cells, and that would be its normal function. And this function might get corrupted in diseases like Parkinson's disease, where it can also help inadvertently spread aggregates between cells. And so our hypothesis is that this protein called PRPC is necessary for alpha-synuclein aggregates and Lewy bodies to spread from one cell to another in the brain, and this is important for understanding Parkinson's disease. And so what we're doing is we're using mice to study this question. So there are mouse models of Parkinson's disease, such as the one that we use called M83 mice. So these are genetically modified mice that produce in their brains higher than normal amounts of the alpha-synuclein protein, and it's a mutant version of the protein that would cause Parkinson's disease if present in a human. And so this is necessary to be able to sort of recapitulate some of the key features of Parkinson's disease, which would occur in people who are 60 and 70 years old within the 2-year lifespan of a mouse. So when these mice get old, they display some of the symptoms of Parkinson's disease. And if you look at their brains, they have Lewy body-like aggregates in them as they get older. So what we're going to do is, there are also mice called PRP knockout mice. So these are mice that have been genetically engineered so that they don't produce PRPC. And one of the reasons why we don't know what this protein does is because when you get rid of it, nothing happens in the mice. And so what we're going to do is, we're going to breed these Parkinson's disease mice with the PRP knockout mice to create mice that have the Parkinson's disease gene and don't have PRP. And we're going to compare the speed at which they develop Parkinson's-like symptoms and pathology compared to mice that have the normal amount of PRPC. And our hypothesis is that in the mice that don't have PRPC, that they'll develop disease much slower or perhaps not at all, because the proteins will not be able to spread within the brain. Why are we doing this? Well, we think that preventing the cell-to-cell spread of alpha-synuclein aggregates may be one strategy for halting the progression of Parkinson's disease. So that if we could block this interaction between PRPC and those alpha-synuclein aggregates, that might help to prevent the spreading of the Lewy bodies within the brain. And we know that this is likely to be a safe strategy for treating Parkinson's disease because, for instance, one of the ways of reducing the interaction is by lowering the amount of PRPC in the brain. And we know that this is safe because if you have no PRPC, you're also resistant to prion disease. And so people have been looking at this question for a long time. So fish, mice, goats, and cows have been genetically engineered to lack this protein, and they seemingly live to old ages without having any major problems. And so it seems that this would be a safe strategy. And a lot of the reagents to do this are already in existence because they have been used to study the prion diseases. So I'd just like to end by saying a big thank you to the Scottish Rite Charitable Foundation of Canada for funding this research, and thank you to all of you for your efforts in fundraising that supports the foundation. And I'd be happy to take any questions now, or if people don't want to ask questions now, I will give out my email address and I'll be happy to answer questions by email later. Thank you.